diagrams of transcription and translation.
With units, like subunits and things.
Yeah, I would know the picture. I would know, like...
The A site and P site?
Yep. I would know where
base,
the mRNA
base codons.
What way it's going in, 5-cap, 3-cap. I would understand where introns are and extrons are.
What
exons are.
Introns and extrons.
Exons, yeah.
Okay.
The parts that we take out and the parts that we keep and glue back together before we send it out of the nucleus.
Okay.
Guys.
There'll be diagrams you've seen before, so just make sure you study what you have.
Okay. The most important function of the electron transport chain is to A. Reoxidize NADH and FADH2. B. Generate a hydrogen ion gradient. C. Phosphorylate ADP. D. Convert oxygen into water. E. Pass electrons to oxygen.
It's either pass electrons to oxygen or an electron gradient. I remember that one was like, I think when me and Miss Feeney went over that one, it was like arguable.
It's
I
B.
said, what was
I
the
said
question?
B. The most important function of the electron transport chain is to generate a hydrogen ion gradient.
Or to get the electrons to oxygen.
What was the other two?
Reoxidizing NADH and
What is the final
FADH2.
process of ETC
Is
Converting
chemioosmosis.
Yeah.
to produce
ATP.
That
So it would be-
your answer.
Okay.
Yeah.
Yeah, no, it
phosphorylate
has
ATP.
phosphorylate ATP. Which that is just like the turbining is phosphorylation.
All the pieces
but
are
it's
important,
called
but the main
chemiosmosis.
function of the ETC is to do what?
Phosphorylate ATP through chemiosmosis.
Okay. Oh, I'm stuck.
Okay, I need to get some work done here too.
Okay, 2.
Almost done marking your first part of your labs.
Oh.
I just fell asleep last night at 12, so I didn't finish some people's.
Which of the following sequences correctly describes the overall process of metabolizing glucose? Okay. A. Pyruvate oxidation, Krebs cycle, glycolysis, electron transport, oxidative phosphorylation. B. Oxidative phosphorylation, Krebs cycle, pyruvate oxidation, glycolysis, electron transport. C. Krebs cycle, electron transport, glycolysis, oxidative phosphorylation, and pyruvate oxidation. Or D. Glycolysis, pyruvate oxidation, Krebs cycle, electron transport, oxidative phosphorylation. Or E. Krebs cycle, oxidative phosphorylation, glycolysis, electron transport, and pyruvate oxidation. I don't even remember what I said.
Dude, it's the order.
Yeah.
It's
I think it's
What comes
It's-
first?
glycolysis.
Good.
Pyruvate
So eliminate
oxidation.
any of the ones that don't have glycolysis.
Krebs.
So D. Glycolysis, pyruvate oxidation, Krebs cycle, electron transport, and then oxidative phosphorylation.
Mhm. Like, if it confuses you, like, go back, reread it, and then eliminate right away the stuff
ones
that
that
you
aren't
know.
right.
Yeah, like,
Yeah.
when you should be able to look at it and easily eliminate at least
Mhm. Yeah.
one or two.
You're okay with us like scratching out the answers on the exam, right?
Yeah, you can write all over it as you want, because I'm gonna mark on the answer key.
Oh, like
We're gonna
this.
do the
Okay.
page beside, like a bubble sheet,
Okay.
and then you put all your 50 there, so you can write on the multiple choice all you want.
Okay.
Remember, use your exam to help you,
Mhm.
right? Because 52 multiple choice, there's answers in there.
Yeah.
So if there's answers in there that can help you answer things in short answer, then use it. Does that
Okay.
make sense?
Yes. 3. Stop looking glum. Energy to phosphorylate ADP during oxidative phosphorylation is delivered from A. The hydrogen ion gradient across the outer membrane of the mitochondria. B. Energy released from a reaction when converting a substrate to a product in glycolysis. C. Energy released when electrons are transferred from one carrier to the next. D. Energy released from a reaction when converting a substrate to a product in the Krebs cycle. Yeah, I said that
Make
right.
sure you really know the difference between oxidative phosphorylation and substrate level phosphorylation. That's the one that I find you guys get confused and don't understand.
Mm-hmm.
Like, what's oxidative phosphorylation?
It's when pyruvate is meeting acetyl-CoA.
Yeah.
Or, sorry, that's pyruvate oxidation. Phosphorylation.
Phosphorylation deals with ATP.
P gaining to ADP.
So ADP binds with P
Mm-hmm.
via the actual like ATP synthase. So you got to have something transferring electrons or something being pumped, like a hydrogen pump that pushes through to oxidatively phosphorylate ATP. Whereas substrate level is sort of the sidebar ones.
The GDP and G.
You know, like the sidebar ones where we have them intermediately within the other pieces.
Okay.
Do you see what I mean? Like we have to have the substrate accepting something, like the molecule in glycolysis, glucose goes through.
Yep.
And you know how a P is pulled off of an ATP and attached to one of the molecules along the way?
Yeah.
That is substrate level phosphorylation. Whereas when we just make ATP using a pump or hydrogen or energy from electrons or somewhere else, that's oxidative. Like
Okay.
think about oil rig. Oxidative has to do with electrons and protons. That's the best way I remember it, is oil rig deals with
I like
oxidation.
Leoker personally, but that makes sense.
Yeah.
No, that makes complete sense.
So oxidative phosphorylation deals with hydrogens and electrons, whereas substrate deals with within the actual steps.
Yeah. Like
where
Krebs,
we're
for example.
Pulling phosphate off of something and attaching it to something else.
Yeah.
Right?
That makes sense.
Oh, I missed one. Okay. I missed two. Here, maybe this one we could put. These are like the silly questions that you like hated reading to me when we were studying for the other tests. Oh, don't forget that. I should have brought my cue cards. That's what I should have done. What type of environment do cells tend to exist in?
Aqueous.
Y'all got that. Is the cell membrane A, impermeable, B, permeable, or semi-permeable? You got that. Define the function of the cell membrane.
Oh, to make sure only certain things go in and outside of the environment using certain facilitated proteins.
Yes. How does the cell membrane maintain integrity?
Through electronegative energy, or like intermolecular energy, like
yeah,
keeping.
like the pathways?
What was the question again?
How does the cell membrane, how does the cell membrane maintain integrity?
Meaning, like, to keep its shape?
Like, maintain its strength and work properly.
Well, then, yeah, it would be like the proteins and, like, you know, the environment, making sure everything's equilibrium, balance.
I love equilibrium. Selective equals. What is selective? Permeability. Only one certain things do. Trust in that. What's impermeable mean?
Nothing can go through.
Structure. What soluble molecules enter the cell easily?
Soluble molecules? I think oxygen. Yeah, oxygen, right?
Maybe.
Is there not an answer on there?
There is.
What?
What's the answer?
Yeah.
Isn't it like lipids?
Yeah, lipids as well as small polar molecules like
Like
potassium and
oxy-
hydrogen.
Oh, okay.
That's okay. A. What forces hold phospholipids together?
Intermolecular forces?
How is this beneficial to the fluid mosaic model?
Keeps like the membrane and like the semi-permeable membrane and some.
I would know the phospholipid bilayer for sure, and all the pieces. I would
Yeah.
know it the way that we answered that communication question. So I know all the pieces to it.
Communication.
Like the
Oh, like
hydrophilic,
when we were in the hall?
hydrophobic parts of the actual phospholipid bilayer, understanding what cholesterol's in there, understanding how certain molecules can diffuse but others can't.
Okay.
And fluid mosaic model, what does that mean?
It's just a model of all the different things, like of the proteins and everything, right? Like the.
Fluid mosaic model means, if you break that down, fluid means
The
constantly moving.
Like the intermolecular force.
Sort of the intermolecular forces keep it
Or the extramolecular
semi-permeable,
fluid?
but also keep it solid so that certain things can't come in,
Mm-hmm.
but certain things can. So when we watch the video of the phospholipids, right?
Like moving.
And they're constantly
Yeah.
vibrating, and we know that cholesterol is doing the same thing. That is the fluidity, and because it's lots of different things in that phospholipid bilayer, that makes it mosaic, because we have phospholipids.
The bunch of different
we have a bunch
facilitated,
of different
yeah.
things that are in there to keep it to be what it is.
Okay.
Does that make sense?
Yeah, no, that makes sense. And cholesterol is what maintains fluidity within the phospholipid bilayer,
Cholesterol, depending on temperature, yes.
depending on temperature, because then it can denaturate. It can be denatured and things like that.
That deals with proteins.
Okay.
So cholesterol will allow for more fluidity or less fluidity depending on different changes.
Like the temperature.
Yeah.
And the fluidity and stuff, right? Because it keeps it together, and then it keeps viruses out when it's too loose.
Yeah, and it just sort
And
of
it just-
hangs out, yeah.
What type of bonds exist between phosphate heads?
I know this.
Is it? It's not phosphodiester's.
No,
It's
it's like
not
a really
peptide.
simple, like.
Yeah, that's why
Not
I know
peptide
it.
bonds.
No, don't think too hard about it. It's just an element, and then it says bonds.
Oh, phosphate bonds or hydrogen bonds.
True or false? Oh. Hydrophobic tails are held together by hydrophobic interactions.
True.
Yeah.
Fire, y'all are so smart. Let's move on to the fluidity of the membrane. If the membrane is too fluid, what does that mean?
No working.
Things can go inside of the cell, like inside of it.
Yeah, so like what's that
It's not-
called? Like,
It's permeable.
there you go. And if it's too rigid,
It's. Oh, that's a good one. I forgot about that one. Is it
it's just the
selective?
opposite.
Impermeable.
Impermeable? Okay.
When the temperature goes up, what happens to it?
The fluidity.
Intermolecular force gets. Yeah.
And when the temperature is too low,
The fluidity
Mm.
decreases
it goes.
and shrinks. And
And then
Yeah,
it, like,
swell
seizes
and then
together, and
shrink.
it doesn't want to do anything in its life, and it just wants to die.
Me.
Oh, no.
We're in Doc Martens.
I am.
Like, I'm in love with
I know I have to,
you
like,
now.
I didn't really want to wear them because I don't really wear them. It's not my biggest style, but I should probably use them.
Okay, so if there's a strong intermolecular force upon it, what does that do to the fluidity?
It would make a strong intermolecular force would
Mhm.
make it shrink, right?
And a weak intermolecular force,
Make it really fluid and swell.
it'll make it dance. Eukaryotic cells What fat is
You're
found in most
eukaryotic.
eukaryotic cells?
What? Sorry.
What fat is
Mitochondria.
found in most eukaryotic cells?
Carbohydrate. No.
Cholesterol? That's not right.
It's
Is it cholesterol?
Is it really?
Yeah, because it sits between the phosphate
Yeah.
heads. What is the function of cholesterol?
To maintain fluidity within the phospholipid bilayer.
Where is it
You got
found?
It is found between the phospholipid heads in the bilayer.
I was like, I just said this. I feel like there's more to that one. It's okay. Active transport. What is it?
It is active transport is when it needs facilitated things like facilitated diffusion, osmosis, things like that. Correct?
You are right, but I just wrote involves moving substances against a concentration grain. You've got that.
Yeah, so it's
right.
facilitated. And then
You said
passive transport is when it can easily just go through the phospholipid bilayer, like hydrogen or potassium that have high polarity.
There you
There you
go.
go.
Identify the 3 types of active transport.
I think I already did osmosis, facilitated diffusion.
Mm-mm.
Oh, sorry. The question?
Identify the 3 types of active transport. The 2 of them start with an E and 1 starts with a P.
Oh, endocytosis and exocytosis.
Uh-huh.
Yeah.
And
Phagocytosis? Phagocytosis? Oh, potassium pump. Protein pump. Just pumps. It's potassium pumps.
protein in the cell membrane. And then identify the 2 types of proteins found in the cell.
2 type of proteins found in a cell are interval. Yeah, is it interval?
Integral.
Integral. And then it was peripheral, and then
Oh, right.
peripheral,
They're here right
recognition, peripheral, and then integral. The 3 main proteins.
Peripheral proteins. ...
They create.
Where are they located?
They're located on top of the bilayer, on top of the heads, and they act as markers for other things like carbohydrate chains
And
and amino acid chains or protein chains. Well, amino acid chains are protein. Sorry?
Is it the bilayer or the membrane? Because somebody says it's on top of the membrane of the bilayer.
they're on top of
It's the whole thing is the phospholipid bilayer, but the membrane is like the bilayer, the outside of the bilayer. So like bilayer membrane, still same thing. I guess they
So
can
if I
be.
said membrane, I wouldn't get it wrong because that's what I have memorized as.
No, you'd probably get it maybe wrong because she might think you'd, right? You'd want to say the phospholipid heads.
Yeah.
You'd want to say on top of the phospholipid heads.
They work as signals or markers for different substances, and they support the shape of the cell by connecting to the cytoskeleton. What is a cytoskeleton?
Amino acid chain or carbohydrates. A bunch of ribosomes, a bunch of cholesterol, a bunch of something.
A network of proteins extending through the cytoplasm of eukaryotic.
A network, an amino acid chain. A network of proteins is an amino acid chain, no?
Okay,
Maybe not.
it's okay. I'll say yes. Integral
No,
proteins,
probably not.
also known as,
Potassium pumps or pumps?
starts with a T.
Oh, transport proteins.
Get it, girl. You got it. Trans.
Trans.
I love when my proteins are trans.
Transport proteins.
Trans.
Transmission proteins?
Trans.
So am I wrong? It's not transport proteins?
It can be,
Oh.
but trans.
Membrane?
Yes. Yes. Where are they located?
in the phospholipid bilayer, and on going in and out the phospholipid bilayer.
Somewhere over there.
How is this possible?
This is possible through facilitated diffusion.
No, how
Are you
is
sure?
it possible for them to be fully through the bilayer?
Because
So
of
that's where they grow.
semi-permeability.
That's what it is.
Because it's I would say the same thing though, because of
It has to do with,
semi-permeability.
Properties.
That just says properties.
It has to do with properties. That isn't it.
Oh.
It has to, like, the phosphate heads are hydrophilic and the tails are hydrophobic.
I get it.
Exactly, because that's where
I
they
know,
go.
I get that, but.
Yeah, you're gonna have to do the answer for that one.
Contain both hydrophobic and hydrophilic properties.
Period.
I didn't understand that question.
I don't think that's gonna be on the test.
Identify the functions of integral proteins.
The functions of integral proteins to let
Yeah.
things in and out of the cell.
Yes, so channels.
Yes.
Like,
Channels
you
that
got
let
that? Yes, yes.
things in and out of the cell. But you said 3.
No, 2.
Oh.
You got that. You got both of them.
What were they?
Acts as a channel, allows substances in and out of the
Oh,
cell.
okay. So you just
You
broke
got
it
that.
up. Okay.
I got a diagram, guys. Do you want to look at my diagram? I did it all by myself.
That's beautiful.
Thank you.
I'd show you mine,
Label
but I
them.
can't.
It's written down, labeled them.
Phosphate head, which is hydrophobic.
Hydrophilic.
Or sorry, hydrophilic. Hydrophobic tails. Hydrophobic tails.
This is where the things go in.
This is a protein gate. Or
It is a
sorry.
protein gate.
Yeah, protein gate. Integral.
Yeah.
Where's our peripheral? That's not peripheral.
Yes, it is.
No, peripheral's like on top.
Cholesterol. I'm gonna call it peripheral right now, okay?
Okay. Peripheral. And then where's our integral?
Your
Or
integral.
sorry, where's our recognition? I apologize.
Yes.
Oh, okay. So
Yes,
where's our
that's what-
peripheral?
A peripheral protein is a recognition protein.
No, there's 2.
They do the same thing.
I think it's mine.
And then this is a carbohydrate chain.
You're a carbohydrate chain.
Okay, for sure.
There's one protein that there's 3 proteins. I know we didn't cover the one very much because it wasn't that important, but it's what builds up the cytoskeleton.
Cellular transport.
I don't blame you. I would.
Identify the 2 types of cellular transport.
I don't think you even get a break.
Identify the 2 types of cellular transport.
Yeah, we're gonna go. I know you guys are going to Ireland.
Passive and active.
We are going
Fact
to
on
Cape
fact.
Breton. Yeah, I know you said you'd done it.
What is
So
my mom
you should-
does on
actually,
Facebook
Jen
right
showed me some pictures too.
I don't know the answer to
Kids
that.
when you guys went out. Yeah.
You did answer
So
that.
we're gonna do that. Go
It's not
down
wrong,
and
is it?
around,
Passive
and
and
then up to the
active?
very
Yeah.
top of like the peninsula and
Yeah.
the point.
Passive. What is it?
So I'd like to
I
go.
love those questions.
What is it?
I think after we.
Okay, yeah. Oh, yes. Okay. Yeah,
That's
yeah.
okay. Keep
That's a
writing some next
recognition,
questions.
right?
No,
I
this
figured
is
it'll have
recognition.
Recognition and then on
Little
top
bit
is
of-
Okay.
There's some similarities.
Yeah, a little
Integral.
Would be
similarities.
good.
because the
And this helps-
recognition has the carbohydrate chain.
Anyways,
The recognition is acts as an anchor or marker for carbohydrate chains, whereas the peripheral acts as the cytoskeleton backbone. I only know that because you had that on a
I think it was on a separate diagram.
you had it on a Quizlet or something like that.
I'm sure they can.
I think it was on a cue card.
Oh, that's what it was.
They're killing each other.
Because I didn't understand that too. I thought there was only 2.
What is it here?
Actually, I gotta drop this off too.
Passive transport.
Random. Like they're just kind of like.
It just goes right through.
And then active.
What is this called?
active
It needs facilitated diffusion.
What is this?
Wait, orange?
collisions.
There you go.
And then active is it's intentional.
Yeah. Okay.
Like facilitated diffusion.
Guys, I'll be right back.
Yes,
Okay,
ma'am.
Can we do
Does it-
photosynthesis?
What?
Photosynthesis.
When?
We need photosynthesis.
Okay.
I'm kidding.
sorry.
No, I'm kidding. Keep going. I'm kidding. I was just joking.
Does it work with or without additional energy?
What? Passive or active?
Passive.
No.
With or without? With or without?
Oh, sorry. Like with no energy.
Okay. I was like, no, we don't need to do that question. Okay.
Sorry.
What direction do things move?
Directions move.
Like what concentration?
Oh, things move inside of this into the cell.
Like high to low or low to
Oh,
high?
high to low.
What happens when temperature increases?
Temperature
They
increases,
die.
the intermolecular forces get really high, and then the
Yeah.
cell will expand, and
Yeah.
well, and
What's that
fluidity will
called?
become too high.
What's that called?
For passive
Denaturization?
transport?
Oh,
It's okay.
I'm not sure then.
The collisions just,
Die down. Or no,
they
they're
go
faster.
faster.
'Cause it's hot.
And then
Okay, so
the
just
diffusion rate.
intermolecular. So I would have got it with intermolecular force.
What the fuck? This just says give an example, and it says breathing in the alveoli. Oh, give an
Yeah,
give an
because
example of active transport. Breathing in the alveoli.
breathing in and then your alveoli taking in the oxygen.
Active. Does it work with or without additional energy?
Active works with additional energy.
What is exocytosis?
Exo is when something's leaving the cell.
When it's outside of the cytosis.
Yes. What is endocytosis?
When it's
Entering.
inside the cell.
What does active transport do?
Active transport allows things to
Moves
go.
things actively.
And what does that do?
Moves things through the cell. Collisions? I'm sorry. That is not going to tell me anything about biology other than reproduction.
What are my hands doing?
Masturbating.
Jerking the chicken.
Pounding. Or squeezing.
What is
Squeezing
squeezing
the
also
testes.
known as? Like
Squeeze.
if there was something, if I was like pushing
Did you put your finger?
Stop!
That was the worst thing you could have done.
Pumping!
Oh, they're
That's not
go
how you
like
pump
this.
things.
Go like something like that. Like yeah.
What's it doing,
Is
Chris?
it pumping?
I'm pumping right now.
I was not going like this.
Holy.
Facilitated.
What does this mean?
That's pumping right there. That's pumping. I know pumping
I
anywhere.
was pumping.
That's pumping anywhere.
Oh my god. Let's break. Get them out of here.
sound like study.
It's not at this
definitely
point.
is.
She's
It's-
trying her best.
A facilitated diffusion. What is it?
Slip diffusion is a pump allowing
No, I was just making fun of you. That
Oh.
isn't what it is.
facilitated diffusion
Yeah.
is a pump.
Oh.
It's a pump allowing glucose through the like big molecules like glucose, large molecules like glucose through the cell.
Yeah.
What is your answer?
Diffusion with protein support. Protein tunnels. Protein tunnels are pumps.
Yes.
It's okay.
I probably should have said protein tunnel.
What molecules
PT
must
in my
travel
chart.
through protein tunnels?
PT.
Big ones.
Sorry?
Big ones.
Yes.
That are
What'd you say?
Starts with a P. What
Polar.
molecules must
What was the question? I completely
What
missed it.
molecules must travel through protein tunnels?
Tunnels, glucose.
Tunnels.
Large molecules like glucose.
Yes.
are
In which direction do things travel?
High to low.
You just fucking said that to me and thank you.
High to low. Oh yeah, you're welcome.
High to low. Good boy.
I yet to have a
Most
text,
important-
Christopher.
What?
I yet to have a text.
You get a text?
Oh yeah, whatever, it's fine. Okay.
Most important example of
Oh, you don't even have your ringer on.
facilitated diffusion.
That's why you don't text me.
Most important example of facilitated diffusion.
Glucose going through the cell, through a protein
Going through the
tunnel.
what?
The protein tunnel.
No,
Glucose going through the protein tunnel
like
in the
body,
cell.
Like in your body.
Oh, okay.
Like,
Facilitated diffusion.
like glucose traveling through what?
Glycolysis.
What?
Glycolysis through cellular respiration.
So facilitated diffusion?
Coming in with my breath.
They set the blood plasma into the cell.
Oh, please.
Osmosis is blank, guys. Don't fear.
That's good because I don't know anything.
Oh, water moves in and
Just
out,
a bite.
and out of the cell by what?
Okay.
Is this bitch serious? No.
Get out of my house, bro. Water moves in the cell by out.
The water moves in and the cell by out.
Through osmosis.
In and out of the cell by.
Osmosis.
Which of the following is not a function of protein in the cell?
Those
Element-
popsicles?
Oh,
Yes.
Sure.
Oh my God, you're gonna have to fight over the grape if someone will.
I
want
hate
orange.
grape.
Oh,
I'll take an
I'll have
orange.
orange, please.
Oh, sweet, then we
Thank
didn't have
you.
to fight over anything.
Thank you, Miss Spring.
Guys, this question says, which of the following is not a function of proteins in the cell membrane? And I just wrote none, lol, stupid. I don't even know what the options were.
It's probably none of the above. What were the wait? There was no option. Oh my.
No, it just says none,
None,
stupid.
Wow.
10, like,
0 out of 0. Sorry. What are you doing?
You know, a science teacher just gave us something to eat in a science room.
Hmm?
You're not allowed to eat in a science room.
y'all are fucking weird. Why didn't you break it?
I was trying, but it didn't work.
Okay.
This is not what I needed.
Okay. Glycoproteins consist of
No.
a protein attached to a.
What?
Glycoproteins consist of a protein attached to a.
Phospholipid. Phosphate.
Pinar.
Phosphodiester bond.
Are you biting into your popsicle?
Yeah.
How does that not hurt your teeth?
My teeth are sensitive. Watch.
It hurts my teeth. I can't do it with my front ones. Eat dirty at me.
That's funny. Okay, let's go.
Let's ride.
Glycoprotein.
What's a glycoprotein? I forgot.
You're made of out of them.
Aren't we all?
Not me, obviously. A protein attached to a carbohydrate. The head of a phospholipid molecule is blank due to the presence of the blank functional group.
What the fuck?
It's hydrophobic due to the it's hydrophilic due to the hydroxyl functional group. Or phosphate functional
I
group.
was like, what are you talking about?
Not my thing.
I love you. Phosphate!
Does it hurt my feelings?
Phosphate!
Oh,
Sorry.
stop it.
Okay.
I'm gonna start hearing that in my dreams.
Phosphate! Phosphate!
My sleep paralysis David's gonna crawl on me and yell phosphate in my face.
A phospholipid molecule
Phosphate.
consists of a tail made of.
Hydrophobic.
Great.
I know about hydrophobic, I'm homophobic.
To what?
2 hydrophobic tails.
What else are they called?
Slingshot.
What? Slingshot?
don't-
Fatty acids, maybe?
Oh, fatty acid chains.
You're fatty acid.
I'm so
I didn't
retarded.
like that.
That was so dumb of me. I should have known that.
The cell membrane molecules organize themselves so they are touching.
Head to head.
Good boy. And then when I touch.
I've been called a good boy 4 times now.
Sorry.
The phospholipid bilayer is.
Is that fluidic mom? Antifluoresceic model.
2 layers of phospholipids.
Oh, well, you should have talked about the structure. You said, what is the structure of the phospholipid bilayer? And I would have said the
Well, you should have just known.
Oh, right.
You're just gonna walk
The
into
cell
that
membrane
fucking
is-
cotton mill.
I know. I was going to say, like, that pissed me off. Anyway, let's go.
Yeah, no.
Anyway,
The cell mem- Are you high?
You can't
You should
fucking
be here
here
right now.
I'll fucking say where I want.
The cell membrane is A. Permeable, B. Impermeable, C. Semi-permeable. Okay, sorry.
Guys, that was the first one to eat mine.
Recognition proteins are.
That was the first one to finish.
Normally I am, but it's okay.
Okay.
Recognition
Recognition proteins function.
are attached to.
Carbohydrate chains. Amino acid
Oh,
chains.
that's right.
It's a carbohydrate.
you're right.
It's carbohydrate.
The head of a phospholipid is blank because of the blank
I-
functional.
We
group.
just went through this. It's
Okay, well, it's on a different paper.
It's hydrophobic because of its phosphate group.
I'm homophobic.
Me too.
That's what I think about phosphate. Is that it's just hydrophobic. It's homophobic.
You're homophobic.
Don't say that.
Oh.
I'm
The cell
sorry.
membrane.
I'm
molecules-
transphobic.
Okay, whatever. Okay, whatever.
Wait, I got you. Okay, let's go with this. 1 sec. Let's switch over.
Let's go with this.
Which compounds does the electron transport system in plants use as an electron source?
What?
ATP.
Water, oxygen, NADPH, or ATP and NADPH?
Oxygen.
It is.
Oxygen.
It's ATP and NADPH.
I hate you.
Yeah, Chris, is all your fault.
Okay, do you want me to do another one, or do you want to switch back to yours?
Go ahead.
Okay. No, I didn't mean to take over. I thought
No,
it
go
would be
ahead.
funny. Okay. What is the light used for in the light reaction? What is the light used for in the light reactions of photosynthesis?
Energy.
To synthesize NADPH and ATP? To synthesize hydrocarbons? To synthesize ATP and NADP plus? To fix carbon?
And a DHP plus.
Shit.
I'm gonna say it's to synthesize no.
Okay.
Okay, let's see. Oh, synthesize NADPH and ATP. So the no, you were wrong.
No, I wasn't.
Yeah, you said NADP and ADP plus.
And a DHP.
Did you mean to okay, so maybe you did get it wrong. Right. Anyway. Which of the following is a phase of photosynthesis?
No. None of them.
Krebs cycle,
Kelvin.
the Kelvin cycle, photosystem 1, or photosystem 2?
Kelvin.
It is Kelvin.
Huh?
It's Kelvin. You're right. The oxygen given off by photosynthesis comes from water, glucose, carbon dioxide, Rubisco, or ATP.
Can I go with my boobs? What?
The,
What was the question? Rubbish, go.
the oxygen given off by photosynthesis comes from water, glucose, carbon dioxide, Rubisco, or ATP.
Probably water.
It's water.
I'm so scared.
Shut the fuck away.
Which is true for the reactions of the Kelvin cycle?
No.
Occur in the mitochondria, occur in the stroma, are always light
In the
dependent.
stoma.
Stroma
I'm
are
a
always
four-year
light
stoma.
dependent, rely on presence of oxygen, or none of the above.
It happens in the stroma.
It does happen in the stroma. It do happen in the stroma.
Dude, when you're a nurse, you're
gonna-
gonna have to deal with people doing it in the stoma, and then you have to clean out that infection.
Ew, don't tell
Why
me
do
about
people
that.
do it in this?
It's so common. We talked about this one
I
time.
don't want my wiener in a stoma, bro. 'Cause I
You're
gotta
gonna have to deal with
one.
that all the time, and then you being grossed out by Aaliyah's nails. I was like, oh god.
See, I wasn't that grossed out, but it was just like, wow, that poor girl. That was more of the
And
grossness.
then her
Like,
his
all
dad
going
made them
through it.
walk to the hospital.
Mm-hmm.
What?
Yeah.
Oh my
Bitch pulled out her nail beds and she had to walk to the hospital. Did she show you the picture of her decapitated nail?
No, can you show me?
No.
I don't have it, but like her nail, like her fake nail was here, and then her real nail, you could see like the tissue poking out of the bottom of it.
Holy, what did the dog do?
You just stepped on them.
Oh fuck, bro.
What do you mean?
She was putting her hand up to grab the blanket because it was trying to get a bat that was in Cohen's room. So when she was pulling her hand up to get the blanket, the dog jumped down on her nail and got like the corner of the other one. Like she's got a giant bruise around here and it's kind of broken, and it ripped her nail beds out.
Hashtag go Gemma girl.
How
And then- oh,
strong
the nurse?
was that?
The
It's
nurse,
a German Shepherd.
she was cleaning out her nail bed, and then she took non-stick gauze and started going like this onto her bare nail beds. Mind you, all she had to do was like spray it with a little bit of saline,
And
and
then
then
you
just
dab it.
not scrub for dear life.
That poor woman.
Woman.
That poor woman.
Wormin.
Chemiosmosis depends on protein complexes in the thylakoid membrane.
That one.
A difference in a hydrogen concentration gradient or hydrogen concentration between the thylakoid space and the stroma.
Oh, that one.
ADP undergoing a condensation reaction.
Oh.
The action spectrum of chlorophyll or A and B are correct.
A and B are
Remember.
correct.
Yes, you're right. I love how she was like, oh, that one. Oh, that one. And then A and B was an option. Let's see.
Are you calling me fat right now?
Why would I say that? Yes, you're right.
Oh, so you
Zero.
Mama.
Did your teeth hurt?
Me when my teeth are.
My teeth don't hurt.
Why would you spit it on the sink? and
That's
the garbage
disgusting. I'm done. I'm calling the
Christopher,
police.
shut the fuck
I'm
up.
so done right now.
Alright, we should give you guys popsicle.
Yes.
You did.
She's so sweet.
And then somebody spit it in your sink.
It hurt my teeth.
Well, it's not a big deal.
I'm just kidding.
Would you rather me started crying, Chris?
But isn't it ironic that
Probably.
a science teacher gave us something to eat in the science room?
Miss
You
Bray
guys
gives
never-
Let's all find Miss Bray. I'm kidding.
Miss Bray used to give us banana bread during
Oh,
the
I'm
day.
so joking. That is so cute though.
Oh, I'm gonna
I wish I had banana bread during.
You
Miss
were
Sweeney.
in the chemistry exam time.
I didn't get banana bread. I don't think.
Well, if you were in the class during the exam, you would have
Well, I
got banana
definitely
bread.
was, but I didn't get none of banana bread.
Well,
I
did you
didn't
say
get no
no?
banana bread.
Maybe. Knowing me, I
Right,
probably said no.
banana bread is pretty good. Anything she makes, she's a very good baker.
Like, the only reason
I
didn't
make some
cry
pretty
in the
mean
exam
banana
is 'cause
bread.
I had to shove my face full of banana bread. There would have been tears everywhere.
Yeah.
I'm sorry, I didn't mean You're touching my shirt.
Okay.
Big deal, bro.
Which process is considered a redox step? Carbon fixation, conversion of ADPH, conversion
Oh.
conversion of NADPH to ATP plus. Regeneration, sorry, of RUBP, which is Rubisco,
Yeah.
or formation of Rubisco, or none of the
actually, like,
above
react?
processes. Yeah, I have no idea. Stuff is going on. Which process is considered a redox step? So reduction or oxidization.
Reaching for the light.
Hold on, oil girl.
Oil rig.
Gaining electrons.
Yes, or losing
Oil
electrons.
Rig. Sorry, oil girl.
And those
Oil-ger.
the options are carbon fixation,
Yeah.
conversion of NADPH to NADP plus, regeneration of RUBP, or formation of Rubisco, or none of the above. I think it might be regeneration of maybe regeneration of Rubisco.
A conversation between ADP and NADP.
I don't know why you had to make them kiss, Lisa.
Well, yes, no, you're right. You're right. It would definitely be between the energy
Conversation.
carrying molecules.
Because they meet together.
Yes.
I
And
love,
then
You're
I
right.
love whatever's wrong with you, bro.
So yes, and then now for energy carrying molecules, which one is going through an oxidation, which one's going through a reduction?
NADPH is being oxidized, while NADP plus is being oxidized. But if
Oh, yeah, 'cause it has the
NADPH were to
extra
be
plus.
back to that, it would be redox or reduction.
What is a reduction?
It's
It's
reduction
gaining.
is gaining, while oxidation is losing.
And like it has the plus, so it has the
So
electron
if
to
you're
give.
losing electrons, then you are also losing hydrogens, which makes it that.
NADP plus, so NADP plus would be oxidation, and then NADPH would be reduction,
Reduction is
gaining.
oil rig
Remember
is gaining electrons, so it's an
The
oxidation.
heart baby.
No,
Okay.
the NADH like kissed them, like gave it a kiss. How, like that's how I remember, it, guys. I don't know.
Ow. Ow. Ow.
What?
Okay.
Like when we had I don't know who told me that. Me and Miss Launderville, and she used to tell us about like
Thank you.
the reactions. Like decomposition is when they get divorced, and then the double replacement is swingers, like married people.
Why are all these women married?
What are you talking about? Okay, next question.
The water necessary for photosynthesis is A, split into hydrogen and oxygen, is B, directly involved in the synthesis of carbohydrate, provides the electrons to replace lost electrons in photosystem 2, provides hydrogen added to synthesize G3P, and E, none of the above. I think it's A split into hydrogen and
I feel
oxygen.
like it would make sense. Wait a second here. I actually don't remember.
Oh, 12 doesn't have an answer, like on the answer key. So, Miss
None
Feeney,
of the above.
Yes.
number 12,
None of those to do.
the water necessary for photosynthesis
Yep,
is A split into hydrogen and oxygen.
yep, which is true.
Yep.
Oh.
B is directly involved in the synthesis of carbohydrate. Wrong.
Wrong.
C provides the electrons to replace lost electrons in photosystem 2. Wrong. Because it provides hydrogen to allow. Oh. Okay. Well, the next option is provide hydrogen plus added to synthesize G3P. No.
It must be the first one.
That's how it's said. And then E is none of the above. But
Oh,
C and A are right.
We'll read the question again.
A.
A is split into hydrogen and oxygen.
Well, what's the question?
Oh, sorry. The water necessary for photosynthesis.
Is water necessary?
The water necessary for photosynthesis is A, split into hydrogen and oxygen, or C, provides the electrons to replace lost electrons in photosystem 2. It's how the question is formatted.
Let me find that one. Both are right, but I would think that it's going to be the electron one, because that's a better answer. You can't start photosystem 2 without those electrons being.
Because you need the photosystems through
Yeah,
like
need to be
P60
activated, whereas
or
you
P80.
need water split up, but
That allows the hydrogens to go and allow the process through the electrons.
let me pull that up right now. Ooh.
And on the answer key, it's not answered like through your pink highlighter.
Oh, it's not?
Not on the ones that Farron took photos of anyway at this time.
Oh, should be.
Oh, never trust anything Farron does.
That one I think is.
Send this video to someone who needs it.
And 13 also isn't marked, but that
What do you mean it's not highlighted? Should have been.
I can bring it over if you'd like.
I'm just gonna check my notes. for a moment. Check.
What'd you lose?
Paper
I have a copy of it in my Google Drive.
Next question for now, I guess.
Yes. Next question 13. In the absence of sunlight, plants cannot use Calvin cycle due to the lack of ATP, oxygen, NADP+. A and C are correct, and A and B, or A, B, and C are correct. I would say it needs ATP to allow, like, to do, to do the process.
Yeah.
Oxygen
Is
is
obviously needed. Because, like, oxygen, cool beans.
yeah,
doesn't need
oxygen
that.
is needed. Yeah, for sure. It's your eukaryotic cell. And then NADP plus is an energy carrying molecule that
So,
happens
yeah.
within the reactions. So I think it
That
would be
happens in the
A, B, C are correct, but it's not labeled yet, so we'll have to see.
happens between that bioalkaline and a chemical. No, okay, bye, whatever.
It's the electrons for sure.
Yeah.
Yeah.
Are you not DJing?
Because they wouldn't be able to be energized and go through the electron transport chain through P680 through all the cytochromes to P700 to get where we need to get. And you have to split water to get that.
But yeah, you could also argue that you couldn't be able to provide those electrons at all if you didn't use the first step of splitting hydrogen and water, because you need those.
But you don't use oxygen. Oxygen's just going to leave. the
Oxygen's
plant
gonna leave,
cell,
but you need the
and
hydrogen.
the hydrogen won't be pumped until later in the ETC. So you have to have the electrons charged.
I guess because the electrons are being charged through light energy.
So lysis
first.
has to happen of water to get those electrons charged and into the
What
photosystem
paper
2,
is that?
P680.
That makes sense. That would make sense because we saw through the video the first
Yeah,
step was
so
the
if you look at
energy.
If you look at a video of photosystem 2,
I don't know if I have this paper.
We took a picture of it.
I have to do that. I was wondering if maybe we could just go through the practice tests and then we can mark them up on like what's right, and then Then we can all just like go home and study with it.
Yeah, it's electrons.
I need to do that. Okay, it would make sense overall, because you can't really start the process of lysis and things like that, like you said, without that energy activating the cell.
Yeah, so like, if you don't use those electrons, you still have all these hydrogen and oxygens that are there, but what are they doing?
They can't go through
They
the
can't
cell.
do really anything except oxygen be expelled out of the leaf stoma, right? Exhaled out, like pushed out,
Yeah.
and then hydrogens just hang there, but the ETC isn't charged unless those electrons.
Which activates the, yeah, for sure. That makes sense.
I'm gonna draw you guys a picture while we study. This is gonna be us tomorrow. You keep asking questions.
And I have one more question, Ms. Feeney, that's okay. The question is, in the absence of sunlight, plants cannot use the Kelvin cycle due to the lack of, and then it's,
Sorry, say that question again?
sorry.
Well, there's no sunlight.
In the absence of sunlight, plants cannot use the Kelvin cycle due to the lack of. And then A is ATP, B is oxygen, and C is NADPH. And then D is A and C, and then E is ABC, like all of them.
Well, it's not oxygen. Eliminate oxygen.
So it would be A and C,
A is what?
and that's A is ATP and C is NADPH. And you need ATP to process the cycle as well as the energy carrying molecule.
Yeah, you need NADPH, definitely need that. And you
ATP.
definitely need ATP.
But you don't need oxygen because that's what's expelled through the stoma, like you just
That's
said.
in photosystem 1.
Yeah.
But read the question again to me.
In the absence of sunlight,
Okay.
plants cannot use the Kelvin cycle due to the lack of.
Oh, what's produced, it's NADPH.
Yeah. So okay. So it's not ATP.
Because-
It's just
Because
NAD.
what's produced at the end of photosystem 2 and 1.
NADPH.
Yeah, and if you don't have sunlight producing that at the end, will it be able to go to Kelvin?
No, you won't.
No, so
So you need
that's
you
why.
need those throughout. Yeah, because those are like when the electrons are hitting those. Like, and I'm referencing the video when it hits those little photosystems to allow them to move through the electrons. In that process, that those energy carrying molecules are also helping.
At the very end, the energy carrying molecule is being made, so it can go on to.
Okay, and is
Where's
that
NADPH being made?
NADP? As you just said, it would be at the end of the ETC.
Yeah. So, and we do make ATP there as well, but it won't cycle past. You have to get that NADPH in. There could be ATP in there from other pieces of the reaction, but the only thing coming in external is really the NADPH as that energy carrying molecule.
And that's what starts Rubisco and the
Well, you
recycle.
have to have that to finish the cycle, right? When you see Rubisco, it's at the end of your cycle. So Rubisco is taking basically a phosphate, it's breaking off ATP, breaking ATP apart, taking one of the phosphates, attaching it, so Rubisco can then push through to allow for the cycle to happen again to create those carbon molecules. Because carbon dioxide comes in, Rubisco, the enzyme, comes in as well, causes it to go in the first part of Kelvin to create that bump of the carbon from CO2. Remember, it's not just one CO2 that's coming in.
It's three. Well, it's six,
Six in
actually that gets
total.
split off.
Yeah.
In terms of questions like that, will we see having to explain the split in
No.
Kelvin cycle?
No, I might ask you what the importance of Rubisco is.
Just
Is it a cofactor? Is it an enzyme? Is it
be a
important for
cofactor.
ensuring carbon dioxide enters Kelvin correctly? Maybe something like that.
okay.
There probably is a question but it's a choice question if you want to explain the Kelvin cycle or Krebs cycle or glycolysis, but if you don't know them, those ones inside and out, I would probably choose another question to answer.
Mm-hmm.
So
Yep.
I think Calvin, we didn't spend a ton of time on it, so it's gonna be hard. Because the Rubisco, people don't understand that and
This-
how it transfers the energy very well. It's hard for people to understand that.
Yeah,
part.
no, when we went over, I came over, I didn't really understand it at all. It was hard for me.
It's okay. It's all right. All you have to remember is that it's a cycle that keeps going through.
Just like Krebs, just similar to
And
Krebs.
that's one of the byproducts at the end is glucose, G3P.
Yeah.
I would know what G3P is, I would know what GTP is. Don't get them confused. Does that make sense?
Yeah.
Yeah.
I can see people screwing that up.
Ready for question 14, guys? Which is the correct summary equation? Do you want to look at them?
Summary equation?
Yeah.
Oh boy, I don't know. That's not my mojo right there.
Well, we're probably going to see a lot of this on the test, I'd imagine. There's probably going to be equations, like summary equations, stuff like that.
I wouldn't ask you to write them. You might see one, or you might see some
Like a multiple
pieces.
choice question.
Ask me, Chris, ask me.
Well, let's look at them.
I actually can't
I would ask you to write that out.
Oh my gosh, Hannah. You could have just come with me to get them done again. You're ripping them all off. You're no
They're
fun.
pissing me off and they're bad for work.
You shouldn't have gotten them so long then.
I like them.
You liked them. Which is the correct summary equation, Hannah? Glucose, 6O2, 6CO2, 6 water. Interesting. Wait, summary
Well, you
equation
need
for what?
6 carbon dioxide for Rubisco.
Yeah.
And you
then
need.
the water comes out at the end, right? Like after the no,
I'm not sure where water is during this cycle.
the end, right? Because it's like the ETC at the very end.
Both at the very end, we have the energy carrying molecule of any DPH at the end of the ETC. I don't know if oxygen's also being released, is it?
I think it's one of the final things.
I know oxygen, well, we know that oxygen releases during the stroma and the first part of photosystem,
We know that.
like through lysis.
Okay. Yeah.
So I think, I don't know where the 6 water is coming from.
I am liquefaction.
Well, no, because when during lysis we're creating, are we creating
Wait, the last
carbon
one says
dioxide?
plus energy. I don't think energy comes out.
Well, no, those are the last one. No, energy probably wouldn't go
Okay.
out. We need the energy.
Yeah.
So yes, we can mark off A, or sorry, E. We'd need glucose at the end of it, so we can mark off A as well.
I think you can mark off. Maybe you could mark off D because it has oxygen instead of. No, I mean C because it has water then instead of oxygen. And you could also mark off B because it has energy at the end instead of the reactant.
Okay, so we're only left with D,
Yeah.
which is correct on the answer key, but where's the 6
That's just
water? Where's the
Oh.
6 water coming from?
On D it says 6O2.
Yes, 6 carbon dioxide plus 6 oxygen, or 6 water.
No. It
6
says,
H2O.
oh yeah, plus water plus energy
But-
equals glucose and oxygen.
But in the process, like with photosynthesis, where is water coming from?
I don't know.
Like how is
They aligned. There was like star-crossed lava or something.
Miss Feeney? In photosynthesis, relating to the correct summary equation, it's 6 CO2, 6 H2O, energy that goes into glucose and oxygen.
Say it again? Okay, do it again.
Yep. 6 CO2, carbon dioxide,
Yeah,
6 water, H2O,
yeah,
plus light energy, or energy.
yeah. Like that can go over the arrow or it can be before, it doesn't matter.
Oh, okay. And then it goes into glucose and glucose
Glucose.
and 6 oxygen.
Yeah.
I'm just confused on during like the process of photosynthesis where we have to bring in 6 water, like the same as we bring in carbon dioxide. Are they like broken off?
In photosystem 2,
Yeah, through lysis, right?
yeah, that's the water.
Is that making carbon dioxide as well at the same time? Like, it wouldn't be making carbon dioxide.
Well, the 2 reactants that are happening, you have to think it's happening simultaneously and constantly moving. So with Krebs, Calvin, you have CO2 coming in. That's a reactant.
So it needs oxygen? Or sorry, keep going, I apologize.
So on the reactant side of photosynthesis,
Yeah.
we have water.
Yeah.
We know that because we have water plants, but we also know when we look at the system, photosystem 2 has that lysis of H2O. When the light hits it, right,
breaks.
hits P680, and then hits the water to lyse it, split it up as well.
Yeah.
That's the water.
Yeah.
And there's 6 of them when we look at the whole balancing of equations. When we look at it on paper, we're just simplifying by showing it as one. And it makes sense because when we look at Kelvin, we have those 3, 3, and then 6, and then 3, and then 5, right? So really, it balances itself out. So with our water, there's 6 water
To make that 6
in
oxygen.
photosystem
Yeah.
2 to balance. And then we have what else that plants need? Carbon dioxide. Where's that coming in?
Carbon dioxide comes.
What cycle does carbon dioxide start? It's not in the photosystem 2 or
It's
1.
not beta. I'm not sure.
Kelvin starts, look at your Kelvin cycle. What comes into it?
Well, it does come into carbon.
All the things in that diagram that you see. Okay. Here's that little diagram of the chloroplast.
Photosynthesis. I'm pretty sure. Yeah. My iPad's just sort of.
What comes in?
Carbon dioxide.
So that's a reactant
Yes.
to start it. Okay? So this is a reactant.
And it's just like coming in
Water's a
organically.
reactant. So carbon dioxide's just in the atmosphere. It's just absorbed,
Yeah. Okay.
right? So if I put a plant in the cupboard there, it's still going to be able to live for a certain amount of time because it can still function.
Through the Kelvin cycle.
Yes.
Non-light dependent.
And there's carbon dioxide in the atmospheric air,
Yeah,
right?
because that's what we
And then plants will pull it in through the plant leaf,
inorganically and
through the openings, right?
yeah, through the pores or
Yep. So you go back here, right? Or this one, is that the other one? Where's the other
back
one?
a little bit.
So wherever the leaf is pulled apart and shows you the cell wall, right? So a leaf, a wall,
Epidermis.
okay, we want to be able to move in to this piece here by absorbing carbon dioxide in and allowing oxygen to escape. And that's the cell blown up right there, right? In the
Yeah.
cell wall of the leaf. And so carbon dioxide will get in,
To the chloroplast.
and then we know the chloroplast is going to utilize the Kelvin cycle to absorb that CO2.
Okay.
At the same time, we know in the cycle, so you have Kelvin cycle utilizing a reactant of CO2, and we know water is being utilized in photosystem 2.
Yeah.
Following?
Yep.
So reactant, water's coming from the light, photosystem 2.
Yeah. No, that makes sense.
Okay, so you got part A, part B, reactant. We're not worrying about intermediates right now.
No.
NADPH or ATPs or ADPs or any of that stuff. Those are just intermittent pieces. So pure reactants to start things. Light energy, you already had that on the arrow.
Yeah.
And then on the product side, we are creating
The 6 oxygen and
the
then
glucose and the oxygen.
Yeah.
Okay. Are we creating other things? Yes, but they're intermittent things.
Yeah.
Does that make sense?
Yeah, no, that makes sense.
So we're producing
And we make 1 glucose out of the whole Calvin cycle process.
Out of one rotation,
One
you're
rotation.
getting 1
Glucose.
PGAL, okay, which is a G3P, or just say carbohydrate or starch.
Okay.
Does that make sense?
Yep.
Because we have 1 taken off here to exit out as starch in the plant, sugar, carbohydrates, because plants need it to stay alive, right?
Mm-hmm.
You following?
Yep.
And then the rest of it goes back through the cycle so that we can keep going.
Yep. Okay.
And in sort of a roundabout way, if plants are inside of a plant like spinach, lettuce, any sort of leafy kind of thing, let's say, or broccoli, if all these plants are producing carbohydrate starches within it, and then we decide to eat it, then we are taking those carbohydrates in, and then our body breaks it down to utilize in our system as cellular respiration, because it's still glucose. Does that
Yeah,
make
it's a
sense?
different kind of glucose.
Yeah, we just break down
Yeah.
the carbohydrate. We break down the starch into a sucrose or glucose. or
Yeah, because
whatever the body-
glucose is a carbohydrate.
simplest form. Does that make sense?
Yep, that makes sense. Especially from the taking in and nutrient aspect. But yeah, no, that makes sense.
So the back side of photosynthesis is the front side of cellular respiration.
Yeah, I could see because we have to take in what they're like. Yep,
Are you sure?
I-
Makes sense?
kinda. I mean, you can
So
elaborate if you'd like if you want to go.
So really quickly, we just made this a dual way sort of thing. Photosynthesis, CO2, we have 6 of them. We can just make it simple though, right? For now, that's the Kelvin. Plus water, photosystem 2. To start, right?
Yeah.
Through light energy. So light is gonna get all this going here, boom, so that over on this side,
We have the
we
oxygen
produce the
splitting,
oxygen as a byproduct.
and as well as forming that glucose through the Calvin cycle.
Yes, or starch. So then this stays in the plant. Where does this go?
That goes out through the stroma.
Yep, and we also know that this is produced at the breakup of this, right?
Yes.
Remember?
Yeah.
So what happens is this is the product at the end that is pushed out of the cell, out of the plant. Glucose stays in, and that is photosynthesis. But when you look at this, and you go from this way to that way, this way is photosynthesis.
Yes. And the other way is cellular
Yes.
respiration.
And this way,
Cellular respiration.
we don't use light, but when we take glucose in and oxygen,
We're
And
making water throughout the not the Kelvin, the electron.
electron transport
It's
chain,
electron transport
Good.
chain through
For-
complex 4, and then we're exhaling CO2 after cellular respiration.
after Krebs cycle,
Yes.
right? And then we also know that intermittently in this one we produce ATP 36. And really, this does produce ATP, right, here, to be utilized there.
And energy carrying molecules.
Yeah, but do you see how it's balanced,
Yeah.
right? So reactant side, product side. But when we look at cellular respiration, reactant side, product side.
That makes a lot of sense.
Make sense?
That makes a lot of sense. Well, we see that on the test or the exam.
You might.
It would be cool.
Yeah, you probably have already this part and have to explain it.
Okay, that'd be okay.
Does that make sense?
Yeah, no, that makes
Yes.
perfect sense.
We're just not balancing it right now, but you are right, 6, 6, 6 all the way through. Same with cellular respiration. How many glucose do we have in cellular respiration when we have C6H12O6?
That's just one.
Is it one?
It's just one glucose, right?
Well, one when we talk about one going through, but when we balance the equation,
We'd have 6 glucose.
yeah. Does that make sense?
Yep.
Okay. There we go.
You guys feeling good?
Yeah. Hannah, I'm feeling good.
Hannah, are you okay?
Hannah feels sick. My nail came off. Now I need to get nail care. I can't just miss a nail for graduation. Okay, are we ready to keep going?
Yeah. So do you want me to move on to metabolism or translation?
Was that the last question about that?
Yeah.
Oh, then it's up to you.
I figured it'd probably be smart to go on to translation since we just did that. Okay. RNA is responsible for bringing in needed amino acids, providing the genetic blueprint for building the protein chain, initiating the process of translation, or reading and correcting the protein chain as it is built.
That creates the blueprint for a protein chain, doesn't it?
Replication RNA would, I think that's what it would do. I'm gonna get the thing out though. Just 1 second.
Okay.
You know, like the diagram, like the cute little one. You can go through them if you want. So I'm not taking up your time.
Sit down.
How do you feel done?
She was hanging on by a thread. It's okay. Sorry.
You're good.
Okay.
Okay, RNA is responsible for.
You have me call in.
So we can study longer than till 3?
What time do you guys have to go?
Well, we can't be at the school for a long time.
No, I know. You said you had to go to Dollarama though?
I just had to get nail glue.
Oh, okay. Well, whenever you
But
guys
I can
like
also
are.
do that a different day. It doesn't really matter to me.
Well, whenever you guys want to go or whatever. you
Well,
just-
I'll probably be ready to leave here at like the end of the school day, and then I don't know where you guys want to go to study. I don't know if you want to go to your house.
I have to go. My sister's graduating today because she's like did her on like through like the adults kind of thing, whatever.
Okay.
And there's like a graduating thing going on. So I'll probably be like after when we go like 2:30. I'll probably go back home because I gotta get ready for that.
Okay.
And then maybe we can. I'll call later or something and study. I don't know, something like that.
Something like that.
But I think you're right. Let me look at the answer key. I think you're right though. It replicates.
Now I want to rip all my nails off.
Okay. Translation RNA types.
I would definitely know the different stages of transcription and translation, like
Like
initiation, elongation.
and termination.
Yeah, and know them, so that you could describe, like if I asked you for one of them, you could describe it.
Okay.
Not gonna ask you to describe the whole process.
Will it be multiple choice questions
Oh,
or
there's definitely multiple choice on transcription and translation for sure. Stop codon stuff, start codon stuff, anti-codon, codon combos. Knowing which way to read it,
5 to 3?
Okay.
understanding what the 5 cap versus polytail is, why they're there.
Okay.
Why,
I look
what
at
are
that.
spliceomes? Does that make sense?
Yeah.
I would make sure you know the difference between a point mutation and a frame shift,
Oh, yeah, okay.
and what a missense, nonsense all mean. Examples. Does that make sense?
Those will be used in short answers.
It could be, or I could give you a mutation and say, what type is this? You guys are gonna have to use, the amino acid chart.
Yeah.
Yes.
We give you one, everyone gets one, and some of the questions you're gonna have to use it to figure out.
And what we're gonna have to build the protein chain, and then from there we'll say the mutation is so such from the protein chain.
Could, or I might give you all that and you have to find it and figure out, is this mutation harmful or not harmful?
And how would we know?
And is it a stop codon? If it is, then it's a nonsense. Is it an addition or insertion? Is it a deletion? Does that make sense? Remember that note I gave you with all the different types?
Of mutation? I remember the note. I just don't remember.
Too crazy on that.
Okay.
And the we didn't do a ton on the promoter region or the coding at the beginning of the 5 cap, and we basically just talked about the start codon,
Mhm.
stop codon, creating
Translating
a
it.
protein polypeptide chain, right?
Yeah.
Understanding A site, P site, understanding anticodons, codons, and then being able to look at that and go, oh, well, it just changed something in the DNA, so that must mean there's a mutation. Does that make sense? So I would
Yeah.
give you a strand, and then I would say, a mutation just happened in position number 5, where A is now changed to G. What does this do to the protein outcome? So then you would have to go through those steps and show me what happens when you change that number 5 position to the G.
And then in terms of completing the protein chain and everything and showing like the mutation or whatever, then how do you explain what kind of mutation it had or if it's bad or good? Because would that be before
Well, if it's a stop codon in the middle of the DNA, if
That's what I was
mutation
just about to
comes
say,
in
if it's
and
like
it changes
the A to a UGGA or
or something, and
GUA?
it's a stop codon, that's not good in the middle of a protein, because then the protein, is it gonna work?
No.
It will just, it
It's-
won't be functional,
Yeah,
and it'll
and there'll be a
probably
metabolic
it'll
disorder.
just not work.
Yeah.
right? So then you would say that.
Okay.
But if it came in and it just changed an amino acid from gly to tri, it's still gonna make the protein, it might just be functionable for something different.
Okay.
Or maybe the change didn't change the amino acid at all, because some amino acids have 3 combos, right? Like the same as stop codons.
Like FOS and PRO and yeah.
See what I mean?
Yeah. That makes sense. In terms of photosynthesis, what should we know? We know photosystems, we know the
Know the
Kelvin
order.
cycle, and things like
know what's
that.
coming in to start photosystem 2, know what's being produced at the end, know what's going into Kelvin, know what's produced from Kelvin, know where it happens. Does one happen in the cytoplasm? Does one happen in the thylakoid space? Does one happen in the stroma? Does that make sense?
Yeah, and will those be,
Could be multiple choice,
could be short answer,
could be a short
depending
answer.
on. And with short answer, well, when we pick, like, say we know Krebs cycle, and you're talking about explaining like Krebs cycle, Kelvin, blah blah blah, do we just have to explain just one, or do we have to explain all three?
When I say Krebs, you would just say acetyl-CoA is coming in to start the cycle. It is changed into citrate because oxoacetate joins with acetyl-CoA to get it started. And it goes through that cycle to produce so many things along the way, NADH, FADH2, right?
Yep.
ATP. And at the end, we know it's gonna go around one more time because one glucose molecule allows for 2 turns in the cycle. That would be 4 marks. You don't have to explain boom, 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 boom. If you wanted to talk about how we go from a 6 to a 5 to a 4, and then talk about the energy carrying molecules that are being used or produced, and the number of ATP total in Krebs that are produced, and then what's going on to the ETC, that would work too. There's lots of ways to get 4 marks.
Okay.
Just make sure you know the proper things coming in and proper things being produced.
Gotcha. And biochemistry with terms of like lipids, proteins, and nucleic acids and carbohydrates. Knowing that they're like nucleotides and all those kind of, and
Yeah, I know the monomer pieces, know the polymer pieces.
monosaccharides, because
And then-
carbohydrates are built from monosaccharides, correct? Yeah. And then monosaccharides can be monomers, polymers, or it can be made up of.
Monosaccharide is a monomer, which the easiest example is glucose.
Okay.
Whereas a disaccharide is a polymer, which is also a disaccharide, which is a carbohydrate because it's more than one little subunit.
Right. That was a lot
I
in one
think your
sentence.
brain is like burnt right now. I think you need a break from studying. I think you've been going too much study mode. You need a break. You need to go get a Starbucks or something.
I don't think I study nearly enough.
I think you do.
I don't think so, Miss Feeney.
I've been studying like crazy for bio.
You gotta get that Hannah to do more studying. Put her phone away.
Question 2, Fahrenheit, ready?
Probably not. Yeah.
Or I guess, here, let me find the answer key quickly, because that's what I was doing before I got sidetracked.
Because I already did that.
Well, I was going to move on to the next question, but I realized we didn't answer.
Mad at me, which is so surprising because it was Richard that answered the phone.
Richard.
I hate Richard.
And me.
Yes, Lexi. Yes, Lexi. How can I help? I'm just studying biology after school. What?
Okay, well,
No, you're not literally.
let's move on to the other translation one, because that one doesn't have an answer key. We'll find it.
It doesn't matter.
No, we'll get the answer key, though. At least it wasn't in the pictures. Okay. This one has an answer key, though, and that's what matters. Proteins are constructed within the smooth endoplasmic reticulum, the nucleus, the cell membrane, or ribosomes.
I'm sorry.
Proteins are constructed within.
The ribosomes send like how they're supposed to be built, right? No. No, they're made in the ribosome.
I don't think they're made in the ribosomes.
No.
Ribosomes move them, or like,
Yes.
yeah, things like that.
When does not the smooth? What does the smooth do?
The smooth endoplasmic reticulum deals with detoxification,
So
and
do the rough endoplasmic?
I think it would be the Golgi apparatus stores and packages proteins,
I don't want to talk about the Golgi apparatus, okay?
and then the Golgi apparatus sends those proteins to the ribosomes to be sent throughout the like the cell and whatever. But proteins are constructed,
It must be the rough.
but the rough isn't an option. It's only smooth.
I know. Wait.
It's not nucleus.
That doesn't make sense.
It's not cell
It
membrane.
might be ribosomes.
It's either ribosomes or smooth endoplasmic reticulum. But I don't think it's
That's
ribosomes.
detox.
There's one more, it's detox and what? There's one more thing for the smooth endoplasmic reticulum.
Maybe storage?
Is it structure or storage?
I don't know.
Here, look at the answer key.
What did Lexi want?
It's D, ribosomes.
So I drove her to the grocery store, and then her baby started screaming, so I had to hang up because it was making me angry.
It's ribosomes. They're synthesized in the ribosomes. I guess that makes sense. Okay, RNA is responsible for bringing the needed amino acids, providing the genetic message for building the protein, initiating the process of translation, or reading and correcting the DNA strand that is to be transcribed. Wouldn't it be? Yeah, providing the genetic message, reading, because that's messenger RNA, so it's not D. I think you're right with providing the genetic message for building the protein. Does it initiate the process of translation?
That would be, isn't that tRNA?
Yeah, no, you're right for that, because then we need you're right, I think. And then bringing in the amino acids. I don't know what that means. Bringing in the amino acids is like, I think transcription, or like,
I think so.
yeah. It's C, initiating the process of translation.
Are you serious?
Yeah, so initiating the process of translation is not tRNA. It is in fact RNA, or rRNA. A specific amino acid is brought to the site of protein synthesis by the mRNA, the tRNA, the SSBPS, or the rRNA.
I'm so sorry.
Right. Okay. I'll read it slower. A specific amino acid is brought to the site of protein synthesis by the mRNA, tRNA, SSBPS, or the rRNA.
I actually have no clue.
I think it's tRNAB.
Okay.
I was right. It's tRNAB.
Oh my god, Quizlet is sending me emails now.
Study now.
Your streak is in danger. I don't care.
I had a 24 streak
You just got an email.
during biochem.
That's crazy.
Yeah, it was pretty good. During translation, the amino acid must first enter the acceptor site, peptide site, template site, or anticodon site. I think it would need to enter the anticodon site or the acceptor site. What do you guys take? What's your take?
Acceptor site?
Yeah, it's the acceptor site. And the anticodon, like the base pair, is what goes into the acceptor site, right?
I think so.
Because if we look here, this is our base codon, like the three main pairs, base pairs. This is tRNA, right? Or sorry, no, this is tRNA bringing those base pairs into, into the acceptor site. Okay. Amino acids are joined by blank bond into a let me reread it. Amino acids are joined by a blank bond into a polypeptide chain. It's glycosyl bond, a peptide bond, a phosphodiester bond, or a hydrogen bond.
A peptide bond. It's proteins.
The name's right in it.
Do you want to do it?
Do you want me to do it?
Do you want to switch it up?
It's totally up to you.
Sure.
I can't. Initiation of translation. You literally gave this to me at the worst time ever. Oh my god.
Why?
Because I don't know that much about this stuff. Initiation of translation involves ribosomal subunits joining to the mRNA, the unwinding of the DNA double helix, and the addition of SSBPs to the molecule strands. tRNAs entering the sites of protein synthesis, or the met amino acid joining with another amino acid.
What was the question?
Initiation of translation involves. I'd assume the met one, because that's the start of a.
As a start codon.
It's a start codon.
That is the anticodon. Or sorry, that's the amino acid.
Yeah.
But in order to start translation, don't we need that 3 base pair anticodon? Or is
MET?
the tRNA?
What is the, so AUG is gonna be come in
Yeah.
on the mRNA. That'll be the first one. We know that's a start codon.
Mhm.
What, that is a codon.
Yeah.
Then we know it goes, oh, start codon, AUG, and then tRNA brings in which one to match AUG.
MET.
No, that's
No.
attached to the top of the tRNA. That's the amino acid.
It would do the base pair, so it would
What's
be
the anticodon of the base pairs to
U is
AUG?
ATC, right?
No, T's aren't in.
Or
Oh, it would be
What goes with an A?
UA.
What goes with an A?
U.
U.
What goes with a U?
A.
What goes with a G?
C.
The
UAG.
anticodon.
UAC. UA.
That's the anticodon for start. So when you look at your chart.
So it'd be the tRNA. then.
Yeah. So when you look at the chart, when you guys, that little chart with all the amino acids, right? You should see right here. AUG, UAC. UAC is the anticodon to that amino acid. Does that make sense?
Mm-hmm.
Then
It should be
study.
met. This diagram sucks.
Oh.
And the sets to match those appropriately, right?
Okay.
You can cry, but like if we study, we'll know. I also don't know much about translation.
This is the acceptor site and the P site, correct? Because this is coming in through
So
the.
this is the active site, yeah, it's coming in, and this is the P site in this one, and then it kicks it out after it's connected. So what we see is this one's already connected to the green and they're making the chain.
Mm-hmm.
So as soon as yellow, let's say green was in here, go back like two steps.
Mm-hmm.
Green was in here, yellow comes in, it connects it, and then it shifts green out because it doesn't have
Gets
one.
rid of it, yeah.
Yellow moves into that spot now. It's like playing musical chairs.
Okay.
And then this guy kicks in here. And then the P site goes, oh, we're together. Let's form a peptide bond. And then it forms the little bondy boo-boo with the ribosome. And then it goes, oh, we're
Just
formed
keeps
now.
going.
Let's kick you out and move the blue in here. And then another tRNA will come in based on whatever CGU would be. So if you aren't sure what CGU would be, the anticodon would be, what goes with C?
C is G,
What goes with G?
C,
C.
What goes with U?
A.
A, C, G, A.
And then that is the anticodon to that amino acid. But this, you always read the mRNA to find the anticodon in the chart. Don't use the tRNA. Don't use this little spaceship guy. Don't read this
Because
guy.
that's already the anticodon from
Yeah,
the.
it's not you don't use that to code in the chart. You use the mRNA, always the mRNA. Okay, so
Gotcha.
always look at the codes in the mRNA to figure out what the actual amino acid is. Does that make sense?
Yeah.
Yes.
Don't use tRNA. And I will give you guys those charts, so you won't need them.
Ribosomes consist of 60s and 40s subunits. That's the answer, I think. 2 40s, 2 60s, or 60s and 20s. It's 60s and 40s, right? It has to be. I remember seeing it.
Do
you
not
have the
remember.
answer sheet?
What number is it?
7.
It's 60s and 40s, yeah.
I'm so smart. That anticodon for UUA would be A.
U is
AAT,
AA, and then
B, UUT, C, AAU, and D, TTU. So it'd be C.
AAU.
Translation is terminated when A. A stop codon is reached. B. The A site has no amino acid. C. And the A site is empty. D. All of the above.
I think it would.
That would be all of the above,
Yeah.
right? Is that what it says?
Mm-hmm.
Fact on fact on fact. The resulting protein is at a blank level of structure. A. Primary. B. Secondary.
Like the protein chain.
Resulting protein is all it says.
Okay, so I think the protein chain.
A. Primary. B. Secondary. C. Tertiary. Or D. Quaternary. Isn't it tertiary?
It's primary.
Is it primary?
Mm-hmm. And that makes sense because when we were
For what?
talking about for
The
the,
resulting protein.
and I think it means this before.
Sorry, say that again. The resulting
Protein.
in translation.
I think so, yeah.
Is that what? And what
It's
were
it, but the thing is on translation,
Yeah.
genetic code. So yeah, it would be in translation.
What were the questions?
The resulting protein is at a blank level of structure.
And it gives you all four.
Yeah. Primary, secondary, tertiary, and quaternary.
I would say primary.
It says
Yes.
It
primary.
does change
Hashtag-
into.
Frickin' family.
It does change into a
Tertiary.
quaternary later.
Eventually?
Yes, it has to, once it's fully made, but just initially, it's just the polypeptide, right? Because we haven't gone through post-transitional pieces at the very end. So when you look at your note on the steps, and you flip to the back side, it will say post-transitional, transcription. Does that make sense?
Yes. The amino acid corresponding to CUA codon is
Well, that's definitely something you would get in the exam tomorrow.
I have the thing here.
I don't have the thing on
It's
here.
right here. I just
What's
have to go
that
through all this.
You'll have it somewhere faster.
So you're gonna get this, but it won't be,
CUA.
I won't circle everything, right?
Wait, let me guess.
No, you don't need to guess. I'm
I
gonna
just
give
wanna
you
know
the,
if I
oh, you
know
wanna
that.
know. You don't have to
Lou.
memorize that.
LEU.
What are we looking for? CUA.
CUA.
CUA is Lou. Yeah.
Guys, I did it. It's because I was looking
Where's
at
your
it
other
someone
piece?
yesterday.
Oh, that's it. Yeah, so go to
Yeah.
translation.
This is what I was looking for.
Oh my, what is it?
So there's termination,
I'm dying.
and then there's post-translational processing. So afterwards, when it's done, the ribosome, the endoplasmic reticulum and Golgi apparatus will take them in.
It will
and-
package and transport them.
Yeah, and change them into the globular pieces that they need.
I'd hope the answer is D.
But initially, it is just a polypeptide that comes out, a big long chain.
And how, because we talked about like when we looked at the video and stuff, and how when we made like DNA a double helix and how they bonded, that's what's happening tertiary, correct?
Well, not exactly. Remember beta sheets
And
pleated pieces in the coils?
Yes.
That's what's happening, is that when the protein starts like going like this, or there's another one coming in to make that ball of yarn, you have to think that they have amino groups attached to them, right? these structures. They're structurally
Yeah.
have hydrogens or sulfates or nitrogens or different things
Different
all
elements.
around. So if I start winding them around and these different elements are in different spots, they're going to interact in
With
different
like
ways.
Leo Gurr and things like that.
Well, there's going to be different types of bonds. There's going to be hydrogen bonds. There's going to be dipole-dipole. There's going to
Ethyl.
be van der Waal. All the stuff
Oh,
you get. Yeah.
Chemistry.
Yeah, all the chemistry stuff to keep it in that like ball of yarn.
I hate
Are
chemistry.
you ready for this question, Chris? Because I can't give you all the answers.
Oh,
The genetic code is read in groups of black nucleotides.
thank you, Miss Feeney.
One that's cut off. I think this says 3. I think this says 4. And the last answer is it varies according to organism. So I'm gonna hope it's D.
It's B.
3?
Yeah.
Okay. No, the last question was cut off. Her hands were cut off.
Oh,
Her legs were cut off.
her hands were good off. Her legs were good off.
Her legs were cut off.
Her nose was cut off.
Her arms were cut off.
Her tongue was ripped out.
Dude, there's another question. There's like
There's no
2 more questions to that.
No, it's not, Chris. No, it's not, Chris.
Well, 13 is which of the following is not true for the genetic
None.
code?
You get it?
It is
None,
universal, it is redundant, its start
You're
codon
redundant.
also codes for met, A and B, or none of the above.
Did you just say Pythagorean theorem? What?
I'm sorry, what?
No.
MX plus B?
I think you need a break.
MX plus B. That's the Pythagorean theorem. Is that
No,
right?
it's not.
Or, no?
That is
Oh, it's,
not.
no, it's the one, okay. It's the A, B,
Oh,
C.
it's okay. You're okay. Your proteins just got quaternary. It's fine. Hemoglobin. His hemoglobin fell out. His hemoglobin fell out. Wait.
Doesn't even make sense.
Hemoglobin fell out.
His hemoglobin.
There is such thing as studying too much.
Oh, thanks.
I can never. His hemoglobin fell out.
Do you think I've done that, Miss Feeney?
I
Yes.
think you guys might be close to that point.
What would you
Go take a
recommend?
break. I would go take a break. I'm all about chunking. So like once you're like, I got nothing left here, it's just like, ugh. I would go take a break, go do something, and then come back to it.
Okay.
Every time he used the bathroom, his
Every time
hemoglobin
he
fell out.
hemoglobin.
Okay, guys, I think we do need a
No,
break.
listen. I can't do this.
I think you should practice that mRNA and tRNA.
Yeah.
Go back to those practice ones we did, the sheets in class,
Yeah,
and I would practice going through those.
like the homework stuff you gave.
Yeah, where I said, give me the codes for mRNA.
Oh, like doing the transcribe.
Yes,
Okay.
I would go back and take it and try to cover up the answers and do it again.
Yeah, I'm not comfortable with, in terms of genetics, with translation and transcription or photosynthesis. I just don't feel comfortable right there. So
I would make sure you practice transcription, translation.
Yeah.
stuff.
Sorry.
And just before, like in terms of diagrams to certainly study, we talked about the chloroplast, like the leaf, like with the epidermis and everything. Would that be a key diagram?
I could give you just the classic chloroplasts and say, what's this? What's this? What's this?
Like the ring kind of thing, right? With the little dots in the middle we're talking about.
I could give you that one too.
Okay.
Okay. Or I might give you the one that has all the pieces, like
The
what's this coming
epidermis?
in? Carbon dioxide.
Oh,
What's,
okay.
this structure right here where the light's hitting it? The thylakoid. What is this whole jelly bean? The chloroplast.
And that would be
That would be
The
that
jelly bean?
little diagram you have.
the one we were just referring to.
Yeah, the little one we were just going over. I might give you the leaf.
That's a big.
I might give you the leaf. And then I might give you.
I'm
that
expecting the
little-
leaf.
I might give you one of those pieces, yeah. And then there's the other one that shows the whole all the processes of photosynthesis.
I'm study breaking. I'm doing.
No. Oh, but it's that little one right there, correct?
Yep, that one.
I just don't. I have the one on my iPad.
Yeah, so that one on your iPad, I would ask that one.
Oh, okay.
I would know that one.
And thank you for that.
What is happening?
Genetic code doesn't have any diagrams, so I'll be good there, I guess.
Okay, well, I could
Well, I
give
guess we do have
I
the
could
subunits.
I could give you the
A and P.
transcription, like diagram of the DNA unwinding, and saying, what is this? What is that? Where's this happening?
He would.
And then I might ask you, have the diagram of the
What is that? You
messenger.
mean like tRNA and mRNA? You mean?
Yeah,
right?
I might, and then what is that? An anticodon on the end of the
Yeah,
tRNA.
base code. Like, yep.
Okay, I might ask you, what do these three codes represent? And when you look at it, if it's the messenger RNA, you would say codons. Right? The three base pair, the three.
They're codons to be transcribed.
I think it's a special ed, maybe a special ed boy.
I hope so. I'd be kind of concerned if it wasn't. Guys, I hit a seagull on Highway 2 the other day.
You're evil.
On the way to the... Well, I didn't kill it. On the way to the prom venue during the day. I hit a seagull. It like flew into my car. It was trying to get the food on the road, on the highway.
It flew into your car.
It flew down.
Oh
And
my
I thought I
god.
was gonna miss it, and he like hit my headlight, and I was like, and then Dallas was like, "Oh my god, you just killed it!" And then I was crying on the way to the venue,
You guys are adorable.
and he was like, "Why are you crying? It's a freaking seagull."
I have this all on recording, by the way, guys.
They're not
Oh.
excited.
All of this is on recording, so anything you've said has been held against you in court. Hemoglobin.
His hemoglobin fell
Fuck
out
him.
every time he used the bathroom.
You know.
Is that what happened to Jacob?
That's what's gonna happen to Jacob when I finish with him.
Guys, I do think we need a break. I'm
Yeah, I'm
absolutely,
all done.
I'm
Let's
jumbled.
talk about
I'm all
who
done.
we're talking to right now.
I'm gonna gouge your eyes out.
I'm gonna pull off your freaking fingers.
Ew.
So fun.
I start screaming help.
Help me! Help! Help me! The killer's so bad.
I am the killer.
Ew, it doesn't say like that.
This course said you can go down and like draw out messenger RNA and chalk on the
Oh wow, really, eh?
Oh wow.
I could do. You know what? On the way out, we're right by there. I'll go down and do glycolysis because that's something I can do and impress people. Then I'll just look
Or you
stupid.
could do just
I'll
like,
do crabs.
do a nucleotide, right? Do like
Oh,
a base, a sugar.
okay, yeah, and like a triglyceride, we could do that, or glycerol.
No, you gotta do. Can't I kick out?
Oh, your
Sucky,
great
sucky
acronym.
feral men. Woo! Or wait.
Did you give her bonus marks for that? Because you should have.
No, but I got an
I
LOL.
should've, but then I thought that wouldn't be fair.
I guess.
So
Why?
I should have told
Just
everybody.
because everybody else is boring doesn't mean for anything points.
Some people study differently
I
though.
feel like I have to be fair.
Equality, equity.
Oh, I guess so. It's kind of ginger or something.
We wouldn't have been mad though, Miss
You
Feeney.
did spice up my
Marking, yeah.
moment.
That should be taught in school, I think.
To spice up marking.
No, I meant my acronym. It's not sexual. I don't think.
Could you imagine
Like,
like what?
you're not a sucky man.
Oh, thank you.
He's not the only one.
I actually forgot the acronym as soon as she said it. So.
Well, you know what? I'm just gonna like not drive you home. You can
Yeah,
go
you can
home.
walk.
I,
You can
I'm
be late to
actually,
your
I'm
sister's
used to
graduation.
walking. Oh, Miss Feeney, my sister's graduating today. I know it's not super big importance, but she took your bio class because she took her grade 12, because she's 26 now, I believe. Yeah.
Oh, she did the online with me?
Yeah,
Great,
Caitlin
yeah.
Wood.
Oh,
I don't know if you remember her or
yes,
not,
I do.
but she's always talked about it. And I think she had you at one point in person as well. But either way, it was. I figured I'd let you know.
Oh,
Yeah.
that's
She remembers
good.
if you
Yeah,
tried to
actually,
list
I have a
trouble
lot
with
of
that.
adult ed marking to do right now. And like,
I know one time you showed us a slide that you said was from like combining your, so it was interesting.
yeah.
Oh.
I don't know if I'd want to do like this course online the way they do it. Like,
No,
you gotta
when-
teach yourself all that stuff. It's
Well,
intense.
you guys started with genetics, no? It's different
no,
layout.
it starts with biological molecules too.
Okay.
But it's really boring, like, they have like 12 questions that they basically have to answer.
And it's
Like,
all put into like heat, like it's like
yeah, they don't really need to understand Krebs and all the cycles the way you guys do.
well, right?
Yeah, we do way more in depth than they do, so.
Yeah, we were talking about it because she like we bounce off and whatever, and like she would talk about the things and that, and I would obviously like it'd be just like not necessarily different levels, but it would just be like what I mean. It'd be different.
Yeah, it
But
would
yeah.
definitely would be. It's like that, like taking PSK in person versus online. Like, I wish they offered it to you guys online because those that would take it would get so much more from it.
Yeah,
Learn
you saw
way
you
more
talk about
muscle.
that.
Like the stuff we did at the anatomy lab is kind of what you would do in PSK in person. So
Yeah.
yeah, online
You're
to
like a
me
dog in the
sucks. but-
cookies.
It's unfortunate that we don't have those anymore.
Just keep
Do you
going.
know of Bayside and things like that having those courses?
Bayside was gonna offer it. They had 23 people signed up to take it,
But they
and
never
their administration decided to can it.
Why?
They said
23
there
is
were
big
other-
majority.
I know, they said there were other courses that were in greater need of running, and so they canned it.
Interesting.
Ken, what now?
A PSK, which is not kinesiology.
Yeah, can.
It is kin.
Yeah.
Okay.
I had to do kinesiology for Jacob.
You didn't have to do anything.
Well, no, he failed the class, and I got him his credit in one night. Because I emailed the professor, I was like, hey, can you give me one more night to get all my work in? And I did. I stayed up for 12 hours doing all of his work.
So, as I said, you actually didn't have to
Okay,
do
but then he
anything.
wouldn't have got a credit, and he wouldn't be graduating right now. So,
Yeah,
I
that
love
wouldn't be good.
kinesiology.
You need to graduate. Like,
But
It is so important.
if he would have
It
just
just-
like
it
done
makes
his work
it
instead of
just is
skipping every
it's
morning,
so
he
much
would
harder
have
to come back.
kinesiology is online. You just literally
Well, you're
do
just
your
gonna
work.
come into things where you need a grade 12,
No,
where
he
you're
was
like
always
you're better
there,
off
he
because
just
somebody
stopped.
has one, like you know what I
He
mean,
was never
in
doing his work.
competition of a
He was
job
just
or whatever it
off.
might be.
Yep.
We had the
Especially
same
in your
online.
trades, believe it or not.
Okay, what are we doing?
Okay, let's see what we got here.
What would you guys like to do?
I'd like to go to sleep. I'd
Well,
like to stop
Miss
sweating.
Feeney offered a break, and I will gladly take that offer.
I feel like if I leave, I'm never coming back.
Well, I don't. It's already like 2 o'clock, 1:30, 1:40.
Basically, like 2:30, right? I love when you say basically. That's my favorite because it's never actually like similar to anything.
It's only like an hour. for
It's
me.
basically like 5 o'clock. It's 2.
It's basically like 12 p.m. right now.
I think I just realized it.
I need to ask you guys, just be honest with me, did you
Yes.
study for the communication piece?
Yeah, I did a lot of studying for the communication piece
I've
actually.
been really busy. I tried to do like fit it in, but I've also had a lot of family planning to do because my dad's celebration of life is soon. So
I don't think your prom helped. in
no,
terms
it
of
didn't. 'Cause
Definitely
that was
not over the
definitely-
weekend.
Yeah, I almost feel like if you tell the next group, they should do their prom
I was
way earlier.
I was
I-
looking at it, and we did June 7th last year. That would have been perfect if we did it this year the same.
I think you guys should have done it earlier. It would have probably made your lives
You
easier.
try getting a venue for June 7th, mid-wedding season.
Or even May.
Or like, so I was thinking because I didn't think our exams would be so soon because normally they're like the 20th.
It's not a bad date, it just I
And
feel.
It really clashed.
I didn't expect everything to feel so crammed.
That's
and
what I
then-
mean.
Everybody was dawning on me for the date I chose, and I was like, it was the only one that was open, everybody.
It's a fine date, it's just I think you guys had lots of other things going on.
I
Was
think
mine
I
really bad?
asked, did you study for it? Because there's some I think didn't study.
Was
I
mine
definitely
really
did
bad,
some
Miss Feeney?
reading.
Be
I
blank.
haven't finished yours, but Farron, you and Hannah
Yeah.
haven't finished yours. You won't be happy, but
that's,
you got
I,
a 64 on that.
that's better than.
It's better than a
expected
2%
Better.
or a 0
Why are you saying that like someone got that?
because they did.
A two?
Holy,
Or a
what?
zero? Someone got a zero?
It's probably me.
No, it wasn't. It's the person that walked out at 1:45.
Who walked out at 1:45?
Oh no, she can't tell us, but we're gonna find
Was it
out.
Paige? Paige walked out early, right?